Good morning, sir.
Good morning.
Good morning, sir.
Good morning.
Sir, rename option.
Oh, just a minute. Yes, done.
Good morning, sir.
Good morning.
Good morning, sir.
Good morning. Just a minute. Physiology.
Good morning, sir.
Good morning.
Good morning, sir.
Good morning. Oh, okay. This is
IP class on sir.
no, it is neat, not IP. It is neat part, no? Physiology second. Growth and development. It is a neat. Okay, so yes, this is growth curve, no? First phase is lag phase, second log, exponential, third is another term we use in growth. It is absolute growth. Absolute growth. What is absolute growth? Absolute growth is actually, it is total growth, total increase in size, okay, increase in mass per unit time. That is called absolute growth. It is total increase in size or increase in mass in a given time, given period, per unit time. It is called absolute growth. Okay, so that is you in a given time, total growth in the plant, it is absolute growth. Fine. That is absolute growth. Second is relative growth. What is relative growth? Relative growth, it means this is the growth per unit time, per unit parameter of the growth. What is parameter? This is suppose length or width. Okay, the area. That is, these are the parameters. Fine. So absolute growth is just total growth, overall growth in the plant in a given time. That is absolute. But relative is actually there is a comparison between two. So generally relative growth is shown by leaves most of the time. Okay. So relative growth means what? This is you can say uh, uh, it is defined as increase in dry mass per unit time in a particular system, or you can say per unit parameter, given parameter. That is called relative growth. Okay. So relative growth means what? This is growth per unit time per unit parameter per unit time per unit parameter of growth. Okay, that is called relative growth. See, here there is an example of two leaves. Okay, generally it is shown by leaves. Maximum growth is shown by leaves generally. So here, suppose this is one leaf, smaller leaf. Okay, this is the leaf. Another is larger. Hmm. Larger leaf, smaller leaf. Okay. Its area is around 5 centimeters square, and it's 50 centimeters square. This leaf has to grow just a minute. So this leaf has to grow up to 10 centimeters square. Okay. And its initial area is 5 centimeters. Similarly, this 50 centimeter का जो area है, it has to develop it has to grow up to 55 centimeters square. Clear? This is A leaf, this is B leaf. Okay. So in which leaf you will see more growth, maximum growth? Obviously in A, because A has to develop from 5 to 10 centimeters square. This is the area. Okay. But B leaf, it shows same area has to be covered by both the leaves, 5 centimeter only. But here already 55 centimeter square area, and it has to be grown up to 55, from 50 to 55. Okay, so it means what? It has to cover only 5 centimeters square. This is 5 centimeters square. Okay, so both they cover 5 centimeter only. But in this case, it covers or it grows 100%. It shows 100% growth, because this is 5 and just double of the initial parameter is 10 centimeter. Okay, so it grows from 5 to 10, means it shows 100% growth. Whereas in B case, 50. Okay, this 50, it grows up to 55. So actually it covers 5 centimeter only, but it grows only 10%. It grows only 10%. Fine. So A leaf grows fast. Okay. Whereas B leaf growth, it is slower than A. Is it clear? I repeat. There are two different leaves. First is 5 centimeter square area. Second is 50 centimeter square area. Now both have to grow next 5 centimeter. Okay, so 5 centimeter has to grow up to 10 centimeter. Okay, it has to cover, it has to achieve 10 centimeter square area. Similarly, 50 centimeter square has to cover next 5 centimeter, means 55, up to 55 centimeter square area. So it means their initial parameter is same now. It is 5 centimeter in A case, in B case it is also 5 centimeter. So both have to cover same area, but the 5 centimeter square 5 centimeter ka area is already present in A leaf. It has this 5 centimeter, and it has to grow next to 5 to achieve total 10 centimeter square area. Fine. It means it has to grow fast to cover that area. So it shows 100% capacity, 100% growth. Okay. Whereas in second case, in B case, it has to cover 5 centimeter. Already it has 50 centimeter, and it has to cover up to 55, means only 5 centimeter. That is 10% of 50, 10% of initial parameter. So it grows slower. Okay, iska growth slow aega. So the growth rate is slow. That is relative growth. That is relative growth. Means you are comparing two different leaves, growth in two different leaves in a given parameter. Now is it clear?
Yes, sir.
Are you there? Okay, that is a relative growth. Fine. This is relative growth. So smaller leaf, it shows more growth to cover this same area, whereas the larger leaf shows slow growth to cover the same area. Their target is 50. Their target is 5 centimeter only, na, to grow up to next 5 centimeter. Fine. But in case of A, it shows fast growth and it is 100%. The 10 is just double of this 5, na. So यहाँ पर 100% growth आएगा. And in 50 to 55, it is slower. It is only 10% growth. So that is called a relative growth in a given time per unit parameter of growth. So here parameter is this 5 centimeter area. Okay, fine. Next term, we use differentiation, de-differentiation, redifferentiation. Okay.
Sir, one scroll up, sir.
Mhm. Okay. Do you have that that reso sheet of plant physiology second?
No, sir.
We have PDF, sir.
They sent it
Sir, they
yesterday
sent
in
it
group,
in
sir.
That is a soft copy, I think.
Yes, sir. Soft copy.
Yes. Because abhi hard copy is not available there. Okay. Fine. So you have that soft copy, no? Fine. First. Second thing, you have no test on Saturday, coming Saturday or Sunday. Means need test, okay? Because IP is going. So because of this IP, no test on Sunday or Saturday. This Sunday or Saturday. It will be on next. I think it is 26th or
24th, sir.
24th, I think. 24th, I think. Saturday.
Yes, sir. 25th is Sunday.
Yes, yes. The
24th.
syllabus. Yes. Okay. So we'll cover this growth part. We'll have next three classes, no? Yes, we'll cover this part. Okay. Is it completed?
So should we draw the diagrams also?
If you want, otherwise there is no requirement. Okay. They ask directly this diagram in the examination because the definition is not important here. Okay. So they will give you this example directly. It is given in NCERT also. Okay. So they ask the question on the basis of this diagram, which shows fast growth, which shows 100% growth like this. Okay. Or which is the example of, which diagram is example of relative growth. Next term is differentiation, de-differentiation, and redifferentiation. What is de-differentiation? Sorry, what is differentiation? First of all, differentiation means meristematic cells divide continuously. Okay, and after a certain period, after some mitosis, 2-3 mitosis or 10 or 20 mitosis, whatever the number. So they divide continuous, and finally these cells stop division. These cells stop division and they get differentiated, they get matured. Okay, they get matured. So they get matured means what? They perform a specific function. They start their function, specific function. Okay, that is called differentiation. Okay, they attain particular size, particular shape, and they start particular function also. So that is called differentiation. Like primary xylem is formed, and this primary xylem start ascent of sap and all these things. Fine, that is differentiation, maturation. Next, it is de-differentiation. What is de-differentiation? Actually, de-differentiation means if this mature cell which is not dividing now, because mature cell, differentiated cell have lost their capacity to divide. So occasionally they regain their power of division. Okay, and they start division. So these dividing cells means what? It means they become meristematic again. So these dividing cells, they start division again, and that is called de-differentiation, means breaking of differentiation. Okay, this is de-differentiation. Third is redifferentiation. When these dividing cells, okay, which are de-differentiated, so these living de-differentiated cells start division, and again they lose their power of division. Again they get differentiated, again they get matured cell. So it is called redifferentiation. Okay, see, this is first term, differentiation. Okay, differentiation. What is differentiation? It's very easy. See, just a minute, huh? Before this, differentiation, just a minute. I will show in diagram. This is cell 1. divides into 2 by mitosis. Simple. Again, it divides by mitosis. 4 cells, 8 cells, 16 cells, 32 cells. Okay. After a certain period, after some mitosis, these cells finally they lose their power of division, and now they get mature. Means they do not divide now. Okay, so they get mature. They're in particular size, shape, and they are ready to perform a particular function, specific function. Okay, so this part, that is differentiation. Here, this cell, these cells, it shows differentiation. Differentiation. Okay, this is differentiation. Cells divide, matured, that is differentiation. That's it. Fine. Now what happens? This is permanent tissue, na? This is primary permanent tissue. It is rough idea, just say this one. Okay. So primary permanent tissue. Now, occasionally or when required, secondary growth occurs or sometimes injury occurs. Okay. So at that time, these cells, they regain their power of division. Now they become dividing cell. Again, they are active. Okay. And they start division. Like this. Okay. So again, they start their division. So from this differentiated stage to this stage, again they start division. So it means what? It means you are breaking differentiation stage. Okay, that is called de-differentiation. De-differentiation. It is de-differentiation. Is it clear? This is de-differentiation. De-differentiation means differentiated cells which have lost their power of division.
Sir, can you scroll down?
If they start division, okay, that is called de-differentiation. It is just like plasmolysis, deplasmolysis. Okay, plasmolysis means what? What is plasmolysis?
The shrinking of the cell when kept in hypertonic solution.
Hypertonic solution. Good. So water moves out by exosmosis. So shrinking of protoplasm, that is called plasmolysis. Fine. Now what is deplasmolysis?
When the shrinking cell is kept in hypotonic solution, it
Yes,
regains
that is
its
reverse
shape.
reaction. Good. That is the reverse, no? Just reverse of plasmolysis. So if you are, if you kept that, if the cell is kept in that solution, pure water, so water moves inside by endosmosis. So it means what? It is breaking of plasmolysis, and it is called deplasmolysis. Similarly, here you are differentiated all these cells. Okay. Now these cells, they are divided. Means you are differentiating these cells. So they are matured cells. Now these matured cells, if they start division, so you are breaking this differentiation. No? It is called de-differentiation. That's it. Okay, fine. So these are different. इनको हम बोल सकते हैं secondary meristem. Technically, they are secondary meristem, but here it is not important which is secondary or primary. So these are
So
dividing
once
cells.
can you show last part of relative growth, sir?
This is the relative growth. I will show you full part.
Okay, sir.
Relative growth up to this. Okay.
Yes, sir.
So done.
Yes, sir.
Okay. So what was I saying? Differentiation. De-differentiation So dedifferentiated cells are dividing cells. Okay, these are secondary meristem actually. Now, after some division, suppose this another division, and finally these cells again they lose their power of division. Okay, now they have no capacity to divide. So this is stage that is called redifferentiation. It is redifferentiation. Is it clear? This is differentiation, redifferentiation, differentiation, dedifferentiation, फिर से redifferentiation. Redifferentiation means again the cells after dedifferentiation, the cells divide, and again these cells they get matured stage, they get differentiated. Okay, they attain a particular size and shape, they do not divide, and they are ready to function. They are ready to perform particular function, specific function. So that is secondary permanent tissue. It is secondary tissue, permanent tissue. Fine. That is called redifferentiation. Is it clear?
Yes,
So
sir.
these are three different terms: differentiation, dedifferentiation, redifferentiation. Okay, first term is different. Okay, neat pattern is completely changed now. Neat pattern. They changed this one completely. And it will be in 13 languages. Okay, 13 languages they added: Punjabi, Malayalam, Telugu, other languages. In section A, it will be 35 questions. In section B, 15 questions will be there. Okay, so out of 15, you have to attempt only 10 questions. You have to solve only 10 questions in B section. In A, all questions should be attempted. Okay, so overall 45 questions will come, final figure. And their marking system is same. Okay, positive means correct answer, it gets 4 marks, otherwise -1. Fine. So it will be more easy than previous system.
Sir, only for this year or next year's also this pattern?
Maybe they will continue this pattern. They will continue.
So sir, from now every subject will get 50 questions.
Now every subject, same part. Every subject has 2 sections, A and B: physics, chemistry, botany, zoology. Okay, 35 plus 15. Out of 15, you have choice in this B sections. Out of 15, you have to attempt only
What is
10,
that?
10 questions. Okay,
Sir,
this
what
is
is
new pattern of NEET.
okay?
Exam pattern.
Sir, for 2023 also,
They will continue this part. Okay.
how many questions, sir? So
45, no? This is 45. Overall 45. Actually, they will give 35 plus 15, 50 questions. Okay. But final figure will be 45 only. Into 4. It is 180. Okay. So it will be for 140 and it is for 40 marks. Okay. Means out of 15, only 10 questions. Means you have this choice. Okay. You have to solve 10 questions out of 15. So they changed this one yesterday. Okay. Differentiation. Definition. Meristem divides repeatedly to form new cells. Okay, it divides and forms new cells. After some divisions or some mitosis, the cell it forms this. So after some divisions, these cells become matured and attain their maximum size and shape. These cells are now ready to perform a specific function. So overall, this process is called differentiation. Okay, that is differentiation. So differentiation means cells divide after sometimes they get matured for a particular function. It is differentiation. That's it. So meristem divides to form primary permanent tissue like primary xylem, primary phloem, okay, cambium. So these are permanent tissue. They do not divide now. Now, what is dedifferentiation? Next is dedifferentiation. Dedifferentiation. Just a minute.
Sir, please scroll up.
Hmm. Just okay. So dedifferentiation. In dedifferentiation, what happens if one more point I have to add here. The process is called differentiated. Just a minute. Huh? Add one more point in differentiation here. The differentiated cells I'll show in another color. The differentiated cells have lost their capacity to divide. Okay. At this point in differentiation. Now de-differentiation. What is de-differentiation? If the differentiated cells already differentiated, if the differentiated cells regain their ability or capacity to divide, they start division. These cells, these means dividing cells. These dividing cells form new cells new cells. So this process means this conversion of differentiated cells into dividing cell. Okay, that is called a redifferentiation. So they regain their capacity. This process of conversion of differentiated cells into dividing cells is called de-differentiation.
Sir,
This is de-differentiation.
this process, like, it takes place naturally or in presence of some enzyme or some hormone or something like that?
No, if any injury occurs, okay, at that time or at the time of secondary growth, okay, naturally it occurs, naturally, but controlled by different hormones or chemicals, obviously.
Yes, sir.
But it is not induced. It is because of secondary growth or injury. So wound hormone is present here. That is wound hormone. Okay, at the time of injury, a wound hormone is produced which stimulates mitosis in these permanent cells. So that is possible. Huh? So this is a natural process. Okay, so this is de-differentiation finally. And the third is a redifferentiation. So in redifferentiation, what happens? Again, these dividing cells, after some time, they produce, means they are, I mean, you can say, again, they lose their power of division. Okay. And they get differentiated again into next cell or next tissue. That is secondary permanent tissue. Okay. Like primary xylem, it is converted into secondary xylem. Okay. So because of the activity of meristem, this primary xylem divides, and the cells which are dividing cells, and they are again converted into secondary xylem. So that is called redifferentiation. Okay, redifferentiation occurs. New cells are formed, again they get differentiated. It is redifferentiation. Dedifferentiated cells divide repeatedly. To form new cells.
So once can you scroll up?
Finally, these new cells
Thank you.
Once again lose their power of division. Power of division. Okay, finally. Differentiated once again. Okay. So these cells which are formed by dedifferentiated cells. Okay. So these cells they divide and then finally they are differentiated again. Means they lose their power of division. Okay. So they are this process is called a redifferentiation. This is redifferentiation. Redifferentiation. Okay. So dedifferentiation, redifferentiation. Fine. Okay. After this differentiation, dedifferentiation, and redifferentiation, another term we use that is plasticity. What is plasticity? Plasticity means this is ability of the plant to produce different types of organs, different types of structures, not organs, different types of structures. Okay, in their life cycle. Okay, if one plant is there, so it can produce different structures in its lifespan. Okay, in response to environmental conditions. If the conditions are different, it produces different organ like this. So according to environmental condition or environmental changes or the changes in habitats, so it can produce different structures to survive there. And this ability is called plasticity. I repeat, this is ability of a plant to produce different structures, different types of structures in its life cycle in response to environment. That is called plasticity. Okay, what is plasticity? This is ability of plant to produce different structures, different types of structures in its life cycle in its life cycle in response to environmental condition. Okay, so this ability is called plasticity. For example, here examples are larkspur, cotton, buttercup, and coriander. Okay, so in these plants like coriander, cotton, generally what happens, they have different leaf shapes in their primary stage or developing stage. That is called juvenile stage. Okay, so in juvenile stage of the plant, they develop different types of leaves. Okay, different shapes are there, but in adult stage it is completely different leaf. The leaf is completely different in adult stage. The best example is buttercup. In buttercup there are two different habitats. Okay, so the plant, if it is means in terrestrial habitat. Okay, so in terrestrial habitat these plants develop broad leaves, thin broad leaves, fine, but they produce dissected leaves in aquatic habitats. So it means it is heterophilous condition. No? They develop heterophilous condition. See, this is the example of buttercup. In buttercup, you will see broad leaves like this. Okay, in terrestrial habitat. Terrestrial habitat, not habitat. Okay, but in water habitat, aquatic habitat, here they produce different types of leaves which are dissected. Okay, different leaves are produced which are highly dissected. Like this. So it is completely different from terrestrial habitat. It is aquatic habitat. Terrestrial habit, aquatic habit. It occurs in buttercup. So it shows heterophily, means two different types of leaves. Okay. It is called plasticity, means the same species, the same plant is here, but it can produce two different types of leaves or structures. Accordingly, means according to that environmental condition. If the environment is terrestrial, they have different leaves, broad leaves, and if it is in water, it shows dissected leaves, long ribbon shaped or thin needle like dissected leaves to avoid that water current. Okay, so this specific phenomena, that is plasticity. Okay, That is plasticity. Is it completed?
Yeah.
So this is completion of one part of growth. I will repeat all this portion. First of all, plant growth and development. So you will see growth means what? It is irreversible, positive change, change in size, change in volume, and change in number, not change in number, increase in number. Okay, in case of unicellular organisms like prokaryotes, like protozoans, so they show increase in number. That is also considered as growth. Vegetative growth, what does it mean? It means vegetative part like root, stem, leaves, they show the growth. That is vegetative growth. If stem and carpels, they show growth, means the growth of flower. It is reproductive growth, development of male parts and female parts, stamen, carpels, androecium, gynoecium. Limited growth means after a certain period, the meristem is completely dead, or you can say the meristem is converted into mature cell. So no growth after a certain period. So leaves, fruits, flowers, they show limited growth. But unlimited growth is shown by root tip and shoot tip, that is root apex and shoot apex. Here apical meristem is always present and it is dividing cell, means these are dividing cells, so they divide continuously. That is unlimited growth. Obviously it is slow but continuous. Primary growth means primary meristem, that is primary meristem produced by embryonic meristem of the plant. These are dividing cells, they divide continuous and they form primary permanent tissue like primary xylem, primary phloem, okay, in the leaf, in the stem. So these are primary permanent tissue. This growth is called primary growth. So primary meristem divides and differentiated into primary permanent, that is primary growth. Secondary growth, जो primary permanent tissue है, that they have lost their capacity to divide, but occasionally at the time of injury or at the time of secondary growth they start division. Okay, uh, that is the de-differentiation. Okay, in other words, so secondary growth starts, and finally these dividing primary permanent tissue, they are called secondary meristem, and these secondary meristem finally they differentiated into secondary permanent tissue like secondary xylem, secondary phloem. Okay, so that is called secondary growth. Next is arithmetic. Arithmetic means arithmetic number manner 1, 2, 3, 4, 5. That is important. It is slower. Root and shoot shows this arithmetic. LT is equal to L0 TRT, L0 plus RT. Okay. Geometric growth, so doubles the number in geometric manner 2, 4, 8, 16, 32, 64 like this. So fast growth shown by prokaryotes especially and unicellular organisms. W1 is equal to W0 ERT. Okay, this is the equation. Now phase of growth, as you know, this is phase of division, meristematic phase. Here all cells, in this phase all cells they divide continuously, they multiply continuously to increase the number. That is called meristematic phase. You can see this phase in the meristematic zone of the root. Fine. The tip of the root, you can say, after root cap. Now phase of cell elongation. So behind the cell of zone of division, these cells they attain their particular size and shape. Okay, they increase their protoplasm, they increase their volume. Cell wall materials more deposition occurs. So enlargement occurs finally. That is called cell elongation. Now phase of cell maturation. So after this, finally means finally they attain their maximum size, they get differentiated. Okay, and they perform particular function. So that is called cell maturation. It is zone of maturation. Okay, here no division occurs. Growth curve is there. That is, there are three different phases: lag phase, log phase, and steady phase. So in lag phase there is slow growth, very slow growth occurs, and then exponential growth starts. Exponential phase is there. Fast growth is maximum. Stationary phase or steady phase, so cells are mostly differentiated, most of the cells. So here no division or no very slow division occurs. Some meristem is there only. Fine. So overall, this growth curve is sigmoid or S-shaped curve. Absolute growth means total increase, total growth in a particular time, given time, that is absolute. Relative means there is a relation, there is a comparison between two leaves mostly, and they have to achieve that particular size. Okay, same for same area, they grow differently. Okay, so A, it grows fast, 100% growth occurs. In B case, it is only 10% growth. It grows slow. Then differentiation, de-differentiation, and redifferentiation. So differentiation, earlier I said that meristem cells, they divide continuously and they are differentiated. They do not divide now. They have lost their capacity to divide. That is called differentiation. So primary permanent tissue like primary xylem, primary phloem are formed. Okay. De-differentiation, occasionally these primary permanent tissue, they start division. Generally they do not divide. They have no nature of division. But in certain condition, under certain condition, they divide. They start division. So this is called de-differentiation, or in other word, you can say breaking of differentiation. Okay. So differentiation occurs. Now redifferentiation, again these de-differentiated cells which are dividing, they form new cells, and these new cells again are differentiated into secondary permanent tissue. So that is called a redifferentiation. The last term, that is plasticity. Plasticity means ability of a plant to produce different structures in its life cycle accordingly. Accordingly means according to that environmental condition, okay, where it is shifted. So that is called plasticity. Example is larkspur, buttercup, coriander, cotton. Fine. Okay, one more point I missed here. Not anywhere, otherwise it should be in introduction. Okay. uh, Growth is measured, measurement of growth. Okay, so growth is measured by one tool or that equipment that is called oxenometer. Okay, oxenometer, just like manometer for root pressure. Okay, porometer for stomata, frequency of stomata. Tensiometer is there for water, availability of water. Respirometer for respiration. Similarly here, oxenometer. Oxenometer is present. Oxenometer for growth measurement. Okay, fine. Now the next part of growth and development is, that is growth hormone. uh, Growth hormones, or they are called growth regulator, plant
Plant
growth
growth regulator.
PGR, or they are known as plant growth regulator, PGR. So plant growth regulators are just growth hormone or plant hormones. Okay, so these are some substances which regulate or which control overall growth and the physiological actions in the plant. These are hormones. Plant hormones. Plant hormones. Okay, plant hormones are the substances which control physiological activities of the plant. Plant hormones. There are two types. No? So these hormones are divided into two groups. One that is called promoter, growth promoter. Plant growth.
Sir, can you scroll down?
So one is plant growth promoter.
Inhibitor.
Complete.
And another is inhibitor. Okay. Another is inhibitor. So promoters are auxin, gibberellin,
Cytokinin.
and cytokine.
Sir, ethylene will also come in this, no, sir? Ethylene.
Ethylene is actually it is promoter as well as inhibitor. So it is a separate hormone. Okay, it is not inhibitor exactly. It is inhibitor plus promoter both. Some actions are initiated by ethylene, okay, for growth, but mostly it is inhibitor. So some are means sometimes it is promoter, sometimes it is inhibitor. So it is a different hormone. But auxin, gibberellin, and cytokinin, they always promote the action, promote the growth. So cytokinins, gibberellins, and auxin, different auxins. Okay, so these are growth promoter. Plant growth inhibitor. Inhibitors. Inhibitors may generally abscisic acid. It is inhibitor. But yes, ethylene is also considered as inhibitor because mostly it is inhibitor. Most of the time it inhibits the growth. Okay. But yes, some actions are stimulated by ethylene. So here I'm writing separately. Ethylene is generally an inhibitor, but it initiates some activities or some growth activities. In plant. So that is ethylene. Okay, so there are two different categories, promoters and inhibitors. Promoters and inhibitors. Fine. These are two different varieties. Okay. Next, First of all, we'll take promoters. Okay, so promoter is auxin. First of all, auxin will take auxin, then gibberellin, then cytokinin, and then inhibitor में abscisic acid and ethylene. Okay, so auxins, fine. अभी auxins में these are acids. Auxins are generally acids. Okay. First of all, there is an experiment performed by Darwin, Charles Darwin and Francis Darwin. Okay.
7 scroll.
Darwin and Darwin, they performed this experiment and they reported that actually they studied in canary grass. One grass is there, Phalaris canariensis or canary grass, and he reported that the tip of the coleoptile means grass, small grass. So the tip of the coleoptile, it moves or it shows movement towards light source. Okay, so they kept this coleoptile in the box, dark box, and made one hole at one point. They give light here. Okay, one lamp is there or just open part is there. So light comes from this hole in the box. This is the box. Okay, covered box, and this is coleoptile. These are different coleoptiles. So these coleoptile tips, that is, it is showing curvature towards light. It is showing curvature towards light, like this. Okay, from here, this is the light source. Okay, so from here light enters through this hole and curvature occurs. Fine. So this is the experiment done by Darwin and Darwin. Okay, and the grass, the specimen was canary grass. Okay, so first of all, Darwin they performed this experiment on canary grass. Darwin and Darwin, Charles Darwin, Francis Darwin, son of Charles Darwin, Francis Darwin. So Darwin performed experiment on canary grass. Canary grass का botanical name it is Phalaris canariensis. Okay, it is Phalaris canariensis. So they performed this experiment on canary grass. They reported that when coleoptile of canary grass is kept in dark, and one source of light is given from one side only. The coleoptile or the tip of coleoptile bends towards light. Okay, this is the experiment. This is coleoptile. This one is coleoptile. Okay, now this coleoptile, it bends towards light, and this is the source of light. That's it. This is one experiment by Darwin and Darwin. Okay, first time they reported this phenomena. Okay, means this bending towards light, and then Bison Jensen will come, then finally Wendt will come, Kagel Smith will come. Okay, we'll see later. We'll see next part, means next class. Is it completed?
Yes, sir.
So I'm closing this meeting here.
Sir,
Time
can you
is
scroll down?
from where? From the starting. This is starting of growth hormones. Done?
Yes, sir.
Okay. And this is auxin, starting means some historical points related to auxin. Some experiments are there. So we'll continue this part next time. Okay.
Sir, can you show the paragraph before growth hormones?
This one. Plant hormone.
No, so the paragraph before plant hormone.
Before plant hormone. Plasticity.
Done, sir. Thank you.
Okay.
Sir, what is the
Thank you, sir.
experiment name, sir?
Thank you. Bye.
Thank you, sir.
Thank you. Bye, Ajay.
Thank you, sir.
Thank you, Anushka.
Sir, what is
Thank
the
you, sir.
experiment name?
Oh, Vaishnavi. Sharmishta. Any doubt?
Yes, sir. The experiment name, sir. Darwin's experiment.
The name of that experiment.
Yes, sir.
Actually, this is the book actually, Power of Movement in Plant. Okay. In this, they perform this experiment. That is published in that book, Power of Movement. Okay.
Okay, sir. Thank
They
you.
didn't give any particular name to that experiment, but the book name is Power of Movement in Plants. Okay. Where they published this experiment. Okay. So I'm closing this meeting here now. Thank you.